So, if you look at what's happening right now, billionaires are currently spending just, hundreds of millions of dollars on experimental gene therapies.
Oh, absolutely. The amount of money is staggering.
Right. And hyperbaric oxygen chambers, and frankly, these somewhat, let's be honest, dystopian plasma infusions from younger donors.
Yeah, the parabiosis stuff, it really does sound like sci-fi.
It really does. And because of all that, we've been completely culturally conditioned to believe that cheating death, or, at least fundamentally delaying the aging process, is always going to be this ultra expensive luxury product.
Right. A luxury that most people will never have access to.
Exactly. We always picture these highly secretive, sterile laboratories out in like Silicon Valley. But what if the most scientifically validated anti-aging compound on Earth isn't actually locked away in some billionaire's bunker?
Which is the wild part of all this.
It is. What if it's just been sitting quietly in your local corner pharmacy for the last 60 years, and get this, it costs about $4 a month?
It completely shatters the standard narrative, doesn't it?
It really does.
I mean, the assumption has always been that extending human health requires, breaking the boundaries of modern physics and biology with cutting-edge proprietary tools.
Right. The stuff you can't afford.
Exactly. But the reality, however, is grounded in a cheap, generic molecule that has essentially just been hiding in plain sight.
And that brings us to the focus of today's deep dive. We are looking at the groundbreaking March 2, 2026 publication of the TAME trial. TAME stands for Targeting Aging with Metformin.
A really clever acronym, by the way.
Yeah, they knew what they were doing there.
Yeah.
So our sources for this deep dive include a comprehensive medical review of these freshly released trial results, alongside a highly detailed analytical script that breaks down the actual cellular mechanisms of longevity.
And we truly need both of those perspectives to grasp the magnitude of this. I mean, the clinical data from the TAME trial shows us the physical outcomes, right?
Right, the real-world results.
Yeah, the proof that it works on a population level. But the cellular biology is required to understand the underlying mechanics of why a 60-year-old molecule can actually manipulate the trajectory of human decay.
Which is just a crazy phrase to say out loud.
Yeah.
Human decay.
It's intense, but it's accurate.
It is. So the mission for our deep dive today is entirely focused on you, the listener. We are going to unpack whether a cheap, generic diabetes medication has actually cracked the code to human aging.
And it's a big claim to investigate.
Massive. We're going to explore what this means for your own biological clock, and most importantly, how you can apply these cellular secrets to your daily life without ever even visiting a pharmacy or, getting a prescription.
Which I think is the most empowering part of all this data.
Definitely. Okay, let's unpack this, because to me, this discovery feels like, well, think of it like finding out that a generic rust-proofing spray you've used on your car for decades actually rebuilds your engine block while you drive.
Oh, that's a great way to put it.
Right. It retreads your tires, upgrades your transmission, all while you're just driving down the highway. It's a total shock to the system. You thought it was doing one very specific, rather boring job, and it turns out it's performing a systemic miracle.
That mechanical analogy is incredibly fitting because for decades, the medical community viewed metformin as a single-purpose tool. I mean, it lowered blood sugar for people with type 2 diabetes. That was it.
The boring job.
Exactly, the boring job. But what we are discussing today represents a total paradigm shift in how science views aging itself.
Yeah.
For centuries, medicine has treated aging as an inevitable, untouchable decline, like a law of nature that you simply have to endure.
You just get older, things break, and that's it.
Right. But what the TAME trial solidifies is this shift toward treating aging as a malleable biological process. It is a series of cellular events that can be manipulated, slowed down, and actively managed.
So to understand how we got to this point, like how cheap diabetes drug became the focal point of the world's most ambitious longevity research, we really have to look at what exactly happened on March 2, 2026.
When the TAME trial results were finally revealed.
Right. Let's talk about the specifications of this trial, because I want to be clear, this wasn't some tiny, unverified pilot study done in a basement with, 10 people.
Not at all, no. The TAME trial was a monumental, historic piece of scientific architecture.
Yeah.
It was spearheaded by Dr. Barzilai at the Albert Einstein College of Medicine, and the sheer logistical scale of it is what makes the resulting data so undeniable.
So the sources outline the scope. They used 14 different medical research centers across the United States.
Huge undertaking.
Yeah, and it took roughly 4 years to run, and they enrolled approximately 3,000 adults. But here is the crucial detail, like the variable that changes everything about how we view this drug.
The non-diabetic factor.
Yes. These 3,000 adults were between the ages of 65 and 79, and they were not diabetic.
Which is the defining factor of the entire endeavor.
It's wild to think about.
It is. Metformin has been prescribed to hundreds of millions of diabetics to manage their blood glucose. But Dr. Barzilai's team specifically excluded people with diabetes from this trial.
Okay, so they recruited healthy, non-diabetic older adults and administered 1,500 milligrams of metformin daily, right? Split into two 750 milligram doses.
Correct.
So they gave a blood sugar medication to thousands of people who had perfectly normal blood sugar.
Yes. Because they were no longer testing for diabetes management, they were testing to see if they could intercept the biological process of aging itself.
And the results, the trial revealed an 18% reduction in what the sources refer to as a composite risk, 18% breakthrough.
And that term, composite risk, that's the linchpin here.
How so?
Well, what's fascinating here is that in modern medicine, the standard operating procedure is basically whack-a-mole.
Oh, for sure.
You go to a cardiologist for your atherosclerosis, right? You see an oncologist if you develop a tumor, and you visit a neurologist for cognitive decline. Medicine is deeply, deeply siloed by organ systems.
You never see one doctor for all of it.
Exactly. But the TAME trial researchers understood that aging doesn't work in silos, so they created this killer composite endpoint, which was comprised of 4 massive, distinct age-related threats.
Right. Cardiovascular disease, cancer, cognitive decline, specifically dementia, I think, and overall mortality.
Yep. And when the data was unblinded, the trial proved that the non-diabetic individuals taking metformin experienced an 18% reduction in the onset of all 4 of those massive threats combined compared to the group taking a placebo.
Wait, hold on. I need to make sure we are connecting the dots here because this is huge.
It is.
A drug that only targets the cardiovascular system isn't going to stop a rogue cell from mutating into cancer. And a drug designed to target tumor growth isn't going to stop amyloid plaques from forming in the brain to cause dementia. So if this single $4 molecule is reducing the incidence of all 4 of these completely different diseases simultaneously,
then it proves the drug is targeting something much deeper than the diseases themselves.
Wow.
Yeah, it is targeting the underlying soil from which all of these pathologies grow, and that soil is biological aging.
That's a great way to visualize it.
The drug isn't masking a specific symptom. It is literally decelerating the foundational decay of the human body that allows those diseases to take root in the first place.
That is just a massive conceptual leap. And honestly, looking at the history of it, it makes me deeply skeptical of the origin story.
How do you mean?
Like, how did someone even get the idea to run this trial? Who wakes up, looks at a 60-year-old generic diabetes pill and says, let's give this to 3,000 healthy grandmothers and see if it stops Alzheimer's and cancer? It just seems completely disconnected.
It does seem like a massive leap for sure. But science rarely operates on sudden, unprompted flashes of intuition.
Right, there's usually a trail of breadcrumbs.
Exactly. It usually advances when researchers notice a glaring anomaly in existing data that current models just cannot explain. So to understand the genesis of the TAME trial, we really have to rewind 12 years to this massive observational study conducted in the UK in 2014.
The 2014 catalyst, our sources highlight this as the absolute turning point.
Yes. Now, an observational study is very different from a clinical trial like TAME.
Right. They aren't handing out pills.
Exactly. They aren't handing out pills or controlling the environment. They are simply analyzing massive sets of existing medical records to find patterns. And in 2014, researchers at Cardiff University analyzed the medical records of over 180,000 patients.
Which is just a staggering amount of data to sift through.
It is, but it provides incredible statistical power. So they were looking at 2 distinct groups. On one side, they had patients with type 2 diabetes who were actively taking metformin to manage their disease. On the other side, they created a control group of healthy individuals who did not have diabetes and were not taking metformin.
But wait, if you just compare random healthy people to sick people, the healthy people will obviously live longer, right? Type 2 diabetes takes a terrible toll on the body.
A brutal toll.
Yeah, it shreds blood vessels, it damages the kidneys, causes neuropathy. It's an awful disease.
That is exactly the assumption the researchers were working under. So, to ensure fairness in the data, they used a statistical technique called propensity score matching.
What does that mean in plain English?
It means they didn't just pick random healthy people. They found healthy people who perfectly matched the diabetics in almost every other way. They matched them by age, gender, smoking status, and various other lifestyle factors.
Oh, I see.
Yeah, so if you have a 65-year-old male diabetic smoker on metformin, you pair him with a 65-year-old male non-diabetic smoker who doesn't take the drug.
Okay, so the playing field is entirely level, except for the disease and the drug.
Precise.
So under those rules, the non-diabetics should have won the longevity race by a mile. They didn't have a systemic metabolic disease.
But they didn't win.
Seriously.
Seriously. The jaw-dropping anomaly of the 2014 study was that the diabetic patients taking metformin had an approximately 15% lower all-cause mortality rate than the perfectly matched, healthy, non-diabetic controls.
Oh, wait, let me just repeat that because it sounds mathematically impossible. The sick patients who were suffering from the systemic metabolic disease were outliving the completely healthy people.
Yes. The sheer presence of metformin in the diabetics' bodies more than compensated for the massive biological damage caused by the diabetes itself.
That is mind-blowing.
It pushed their lifespan beyond that of people who had no metabolic disease at all. That was the moment the scientific community experienced a collective shock. They were forced to ask, if this simple molecule is keeping sick people alive longer than healthy people, what on earth would it do if we gave it to someone who wasn't sick?
And that anomaly is exactly what birthed the TAME trial. But this raises another question about the drug itself. I mean, we keep calling it a 60-year-old drug, but where did it even come from? Was it synthesized in some, I don't know, mid-century laboratory from scratch?
The history of metformin is actually quite fascinating, and it grounds it in the natural world, surprisingly enough.
Really?
Yeah, it is derived from a compound found in a plant called the French lilac, or Galega officinalis. Back in the 1920s, scientists discovered that a compound in the French lilac called guanidine could lower blood sugar.
So it started as an herbal remedy. That's wild.
Essentially, yes. But pure guanidine was too toxic for human use.
Oh, okay.
Yeah, it took decades of chemical refinement to synthesize a safer derivative, which eventually became metformin. It was approved in Europe in the late 1950s, though, interestingly, the US FDA didn't approve it for diabetes until 1995.
Oh, wow. That's a huge gap.
It is. So it has this long, deeply established safety profile and a natural origin story, which makes its sudden emergence as a longevity compound all the more compelling.
Okay, so we have the 2014 observational data, we have the historical safety profile, and now we have the 2026 TAME trial showing an 18% reduction in the worst diseases known to humanity.
Right.
But seeing numbers on a page doesn't explain the biology.
Yeah.
To really understand why this is happening, how metformin is actually manipulating time, we need to zoom all the way down into the human cell.
And this requires us to fundamentally separate two concepts, chronological age and biological age.
Because they are absolutely not the same thing, even though we sort of treat them as identical in everyday life.
Right. Chronological age is simply a measurement of time passing. It's how many years you've been alive since your birth certificate was stamped. You have zero control over it.
Right.
Biological age, however, is the physical condition of your cellular machinery. It's the accumulation of wear and tear, genetic errors, and cellular damage.
So in the medical review provided in our sources, they talk about measuring this biological wear and tear using something called DNA methylation clocks. What exactly is a DNA methylation clock? Are we talking about mutations in the genetic code itself, like X-Men stuff?
No, we aren't talking about mutations. Your underlying DNA sequence, the actual genetic code, remains largely the exact same throughout your life. Methylation refers to epigenetics.
Okay.
Think of your DNA as a massive instruction manual. The words in the manual don't change, but as you age, chemical tags called metal groups attach themselves to the outside of the DNA.
Like someone going through the manual with a highlighter or, placing sticky notes over certain paragraphs so they can't be read anymore.
That is a brilliant way to visualize it, yes. These methyl groups change how your cells actually read the instructions.
Got it.
So a gene that is supposed to keep a cell healthy might get covered up by a methyl tag and silenced.
That sounds...
It is. And conversely, a gene that causes inflammation might have its tag removed, causing it to become overactive. The pattern of these tags changes predictably as we age. Scientists, most notably researchers like Steve Horvath, developed algorithms that can look at your specific pattern of methyl tags and calculate the exact biological age of your tissues.
It's like counting the rings on a tree, but at a microscopic molecular level?
Exactly. And the TAME trial didn't just look at whether people had heart attacks or dementia. They measured these epigenetic clocks.
Oh, wow.
The data show that the biological markers of aging, the accumulation of those epigenetic sticky notes, advanced significantly more slowly in the metformin group compared to the placebo group. The drug was actively slowing down the biological ticking of the cell.
So the multi-trillion-dollar question is, how? How does a modified French lilac molecule slow down epigenetic aging? Our sources break this down into three major cellular mechanisms. The first one is called the AMPK trick, which leads to a process called autophagy.
Right. To understand this, we have to look at how a cell senses energy. AMPK stands for AMP-activated protein kinase. You can think of it as the master metabolic regulator, like the central fuel gauge of the cell.
So how does a cell actually know it's starving? Does it have a literal gauge?
It does, actually, and it's driven by a ratio of two molecules. You are likely familiar with ATP, adenosine triphosphate?
Sure. High school biology, the powerhouse stuff.
Right. It is the fundamental currency of cellular energy. When you eat a meal, your mitochondria convert that food into ATP.
Right, and the cell spends that ATP to do its daily jobs, building proteins, moving muscles, dividing.
Exactly. And when a cell spends a molecule of ATP, it breaks off one of the phosphate groups, turning it into AMP adenosine monophosphate.
Okay, tracking with you.
The AMPK sensor is constantly measuring the ratio of ATP to AMP. When ATP is high, the cell knows it is fully fueled.
Because you just ate.
Yep. But when you are exercising heavily or starving, you burn through ATP, converting it into AMP. When the level of AMP spikes relative to ATP, the sensor triggers. The fuel gauge essentially flashes empty.
And what the sources show is that metformin physically interacts with this exact system. It artificially activates AMPK.
It does.
It tricks the cell into believing that ATP is depleted and AMP is spiking, even if you literally just ate a massive meal.
It creates a chemical illusion of starvation.
Here's where it gets really interesting to me, because I love thinking about this through the lens of technology. Think of AMPK as the low-power mode on your smartphone.
I like where this is going.
When your phone's battery drops to 10%, it doesn't just keep operating normally until it dies. The operating system intervenes, the screen dims, background apps stop refreshing, automatic downloads are immediately paused.
It does whatever it takes to survive.
Right. The phone shuts down all non-essential energy draining processes just to keep the core system alive for as long as possible.
That is precisely the biological response. When metformin triggers that false energy crisis via AMPK, the cell immediately shifts its priorities. It halts cellular division because division takes massive amounts of energy.
Makes sense.
It stops building new proteins. The cell transitions entirely out of a mode of, growth and expansion and locks into a mode of conservation and repair.
That triggers the second half of this mechanism, autophagy.
Autophagy translates literally from Greek as self-eating.
Gross, but fascinating.
It is a little gross. When the cell is forced into this low-power starvation mold, it desperately needs energy to survive, but it believes no fuel is coming from the outside, so it starts looking internally.
I picture autophagy as a cellular Roomba.
A Roomba.
Yeah. You turn on the AMPK low-power mode, and the hell says, all right, times are tough, let's clean house. The Roomba wakes up and starts vacuuming up all the toxic gunk inside the cell.
That is a very accurate metaphor. That gunk consists of misfolded proteins, damaged organelles, and specifically dysfunctional mitochondria.
Detract.
Yes. The cell literally builds microscopic garbage bags called autophagosomes around this debris, breaks it down with enzymes, and recycles the raw materials into fresh energy.
Yeah, it's amazing.
By consuming its own waste, the cell effectively renews itself. It cleanses itself of the debris that naturally accumulates and causes cellular dysfunction as we age.
Which connects directly to the second major mechanism our sources highlight, extinguishing inflammaging. Because if the cell doesn't clean up that garbage, the body reacts poorly, very poorly. Inflammaging is exactly what it sounds like. It's the intersection of chronic inflammation and aging.
Okay.
We are all familiar with acute inflammation. You sprain your ankle, your immune system rushes white blood cells to the area, it swells, gets hot, and heals. That is a vital survival mechanism.
But inflammaging isn't a sprained ankle. It's not a blazing fire you can see and feel. Our sources describe it more like a slow, toxic smoke permeating every room of a house over decades.
Yes, remember that cellular debris we just talked about?
The Roomba trash. Exactly. If autophagy slows down as we get older and that debris just piles up inside and outside the cells, our immune system gets terribly confused. Our macrophages, a type of immune cell, look at this cellular garbage and mistake it for a pathogen or an injury.
Oh, no.
Yeah, so they start secreting inflammatory signaling molecules called cytokines constantly.
So the immune system is basically fighting a ghost war against our own cellular trash.
That's a great way to frame it. And it's an evolutionary blind spot. Humans weren't historically supposed to live into our 80s. Our bodies didn't evolve mechanisms to perfectly handle decades of accumulated cellular trash.
We outlived our warranty.
We really did. And this smoldering fire of cytokines damages the endothelial lining of your arteries, which lays the groundwork for cardiovascular disease. It damages neurons, contributing to cognitive decline.
It just wreaks havoc.
It creates a highly toxic environment where cellular mutations are more likely to occur, which leads to cancer.
And the sources show that metformin acts as a targeted fire extinguisher for this exact inflammaging process.
The clinical blood work from the trials confirms this entirely. Metformin significantly lowers key inflammatory markers in the bloodstream. The two major ones tracked in the research are C-reactive protein, or CRP, and interleukin-6, or IL-6.
Okay, I've seen CRP on my blood work before.
Most people have. By activating AMPK and clearing out the debris, metformin essentially cuts off the fuel source for the immune system's overreaction. It clears the smoke, it changes the systemic environment from one of chronic high-alert stress to one of baseline calm.
Wait, if AMPK is the brake pedal that puts the cell into low-power repair mode, there has to be a gas pedal, right? I mean,
Right.
The body has to grow sometimes. Which brings us to the third mechanism, the MTO brakes.
Yes. The cell is constantly balancing between AMPK, the brake, and mTOR, the gas pedal. mTOR stands for the mechanistic target of rapamycin.
Target of rapamycin. Where does that name come from? It sounds like a sci-fi drug.
It is actually one of the most incredible stories in modern biology. Oh, yeah. In the 1960s, a scientific expedition went to Easter Island, known natively as Rapa Nui. They were taking soil samples to look for new microbes. In the dirt of Easter Island, they found a bacterium that produced a bizarre compound with powerful antifungal and immunosuppressive properties.
Just in the dirt.
Just in the dirt. They named the compound rapamycin after the island of Rapa Nui.
Okay, so they find this random compound in the dirt on Easter Island. How does that connect to human cellular aging?
Well, decades later, molecular biologists were trying to figure out exactly how rapamycin was suppressing the immune system so effectively.
Like, what was the mechanism?
Right. They discovered that it was binding to and blocking a very specific protein pathway inside human cells, a pathway that acts as the master controller for cellular growth and proliferation. Since rapamycin targeted it, they simply named the pathway the target of rapamycin, or MTO.
So mTOR is the signal that tells a cell to grow, build, and multiply.
Precisely. When you are an infant growing into an adult, or if you are, say, a bodybuilder trying to add massive amounts of muscle tissue, you want mTORs to be highly active. You want the gas pedal pressed to the floor.
But what happens when we get older? If the gas pedal is stuck down, doesn't that cause a problem?
It causes a massive problem. If mTOR is overactive in an older adult, the cell never gets a chance to rest and repair. It's like revving an old engine constantly without ever changing the oil.
Eventually, it's going to blow.
The cells are pushed to grow and divide until their telomeres shorten and they suffer severe DNA damage. At that point, they should undergo apoptosis, which is programmed cell death.
They should just die off.
Right. But because mTOR is screaming at them to survive and grow, they refuse to die.
They become zombie cells.
The medical term is senescent cells, but zombie cells is highly accurate. They just sit in your tissues, refusing to die, and they secrete massive amounts of those inflammatory cytokines we discussed earlier.
So mTOR feeds the inflammaging fire.
Exactly. Overactive mTOR is a primary driver of aging and cancer, because cancer is, at its core, unchecked cellular growth.
So how does metformin fit into the Easter Island mTOR story?
Metformin acts as a gentle brake on the mTOR pathway. When metformin activates AMPK, the low-power mode AMPK chemically signals mTOR to power down.
Oh, so they communicate.
Yes. It doesn't shut it off completely, which would be disastrous, but it downregulates its activity. It restores a healthy biological rhythm, ensuring that the body spends just as much time in a state of repair and cellular cleaning as it does in a state of growth.
Okay, let's take a breath and synthesize everything we've uncovered so far, because that was heavy.
It's a lot of biology.
It is. We have a generic pill derived from a French lilac that costs less than a cup of coffee. We have a massive multi-year clinical trial proving an 18% reduction in the 4 deadliest age-related diseases. And we have a crystal clear understanding of the cellular mechanics. It manipulates the ATP to AMP ratio to turn on the AMPK low-power mode, which unleashes the cellular Roomba of autophagy, which cleans up the debris, which extinguishes the smoldering fire of inflammaging, all while tapping the brakes on the overactive mTOR growth pathway.
That is the complete multi-layered picture of its cellular efficacy. You nailed it.
So, as a listener, sitting here right now, processing all of this, the cognitive dissonance is incredibly loud.
I'm sure it is.
If this drug is a verified biological clock stopper, if it's unbelievably cheap, and if we have 60 years of safety data from millions of diabetics, why isn't this the standard of care? Why isn't every single person over the age of 30 taking a daily dose? Why didn't my doctor offer this to me at my last physical? Like, what is the catch?
This raises an important question, and it's the most logical question to ask. And the answer isn't purely medical. It's largely bureaucratic and philosophical. The first major hurdle we have to discuss is the regulatory roadblock, specifically the philosophical war with the FDA.
The Food and Drug Administration, how are they viewing this? Do they just hate cheap drugs?
No, it's not that simple. You have to understand how the FDA fundamentally operates and classifies medicine. The FDA relies on standard disease classifications, largely based on frameworks like the International Classification of Diseases, the ICD.
Okay, the rule book.
Right. To run a clinical trial and get a drug approved, a pharmaceutical company must prove that the drug treats or cures a specific, legally recognized pathology.
Right. Heart disease is a recognized pathology. Type 2 diabetes is a pathology. Alzheimer's is a pathology.
But aging is not in the rule book.
Wait, really?
Really. Aging is legally and historically viewed as a natural consequence of time. Because it happens to 100% of the population, the regulatory bodies view it as a baseline biological reality, not a disease that requires pharmaceutical intervention. Therefore, the FDA has literally no established regulatory pathway for any drug that claims to target aging.
So even if a company developed a literal, undisputed immortality pill, they couldn't submit an application for it.
The application would be rejected before the data was even reviewed because there is no bureaucratic box to check.
That is wildly frustrating.
It is, and this is exactly why Dr. Barzilai's TAME trial is so revolutionary. It wasn't just a scientific test, it was bureaucratic jujitsu.
Because they use that composite risk we talked about earlier.
Precisely. Dr. Barzilai and the American Federation for Aging Research spent years lobbying the FDA. They said, fine, if you won't recognize aging as a disease, what if we prove we can prevent your 4 favorite diseases at the exact same time?
A brilliant loophole.
It really is. By proving that metformin reduces cardiovascular disease, cancer, dementia, and mortality simultaneously, they are forcing the FDA to acknowledge that aging is a treatable underlying condition. It is a political battering ram to change the rulebook of modern medicine. So until the FDA officially changes its fundamental philosophy on what a disease is, your average primary care doctor isn't going to prescribe this to healthy people, at least not officially, because it violates standard prescribing guidelines.
That is the current reality. While some specialized longevity clinics prescribe it off-label, mainstream medicine moves at the speed of FDA approval.
But the FDA bureaucracy isn't the only catch here. As I was combing through the sources, I noticed a massive demographic black hole in the data. We keep citing the TAME trial as proof, but we have to look at who actually participated.
Right. The trial was strictly limited to adults aged 65 to 79.
Which creates a massive gap in our knowledge.
It does. If a 35-year-old or a 45-year-old is listening to this right now, and they are tempted to go find an off-label prescription to lock in their biological youth, they need to realize that we have zero concrete long-term clinical data on the risk-benefit ratio for their age group.
We truly don't. I mean, the cellular mechanics of AMPK and mTOR make sense in theory, but human biology is agonizingly complex.
Very true.
Taking a drug that chronically suppresses mTOR and activates AMPK starting at age 30 might have profound unintended consequences over a 50-year timeline. We don't know how it might impact hormone regulation, muscle development, or cellular signaling across decades of use in a young, healthy body. The safety data we have is specifically for older adults or people with a metabolic disease.
And mentioning muscle development brings us to what I consider the absolute most shocking pushback in the entire source material. We're calling it the exercise paradox.
This is a huge one.
When I read this section, it completely upended my understanding of health. We have been told our entire lives that cardiovascular exercise is the ultimate, undeniable longevity tool. Are you telling me that if I take metformin and go for a run, I am actively sabotaging my own workout?
It is a profound and highly counterintuitive paradox, but the data is undeniable. The sources highlight specific clinical studies published in 2019 and 2023 that examine the interaction between metformin and rigorous aerobic exercise in older adults.
And what did they find?
The researchers found that metformin significantly blunted, and in some cases entirely inhibited, the mitochondrial adaptations to the exercise.
The sources explicitly mention it reduces VO2 max gains. Let's break that down for the listener. VO2 max is essentially the gold standard measurement for cardiovascular fitness.
It is.
It measures the maximum amount of oxygen your body can utilize during intense exercise. Athletes will train brutally for months just to increase their VO2 max by 2 or 3%. And this longevity drug stops that progress. How is that physically possible?
To understand why this happens, we have to explore a biological concept called hormesis.
Hormesis, okay.
Hormesis is the principle that a moderate, acute dose of biological stress actually makes an organism stronger.
What doesn't kill you makes you stronger, literally.
Literally. When you go for a hard run or do high-intensity interval training, you are not being kind to your body in the moment.
No, it hurts.
You are creating micro tears in your muscle fibers, you are causing acute localized inflammation, and crucially, you are forcing your mitochondria to work in overdrive, which causes them to leak reactive oxygen species, or free radicals.
So you are deeply stressing the cellular machinery.
Yes. And after the workout is over, your body responds to that intense stress by sounding an alarm. The cells say, that was incredibly difficult and damaging. We need to adapt. We need to build more mitochondria. We need to increase our VO2 max. And we need to strengthen the muscle fibers so that if we have to do that again, it won't be as stressful.
The stress itself is the signal.
The stress itself is the necessary trigger for the adaptation.
But wait, we just established that metformin is a cellular fire extinguisher. It stops inflammation and alters the energy sensor.
Exactly. When you exercise while a high dose of metformin is in your system, the drug artificially blunts that cellular stress response. It scavenges the reactive oxygen species and dampens the acute inflammation. It essentially shields the mitochondria from the stress of the workout.
It's like putting massive shock absorbers on a barbell. You're doing the physical motion of the lift, you're sweating, you're putting in the time, but the muscle itself isn't feeling the full weight.
Exactly.
And because the mitochondria don't feel the full stress of the exercise, they don't send the signal to adapt and grow stronger.
It cancels out the hormetic stress. It is an incredible biological irony. Metformin mimics the cellular effects of exercise by activating AMPK while you are sitting on the couch. But if you combine it with actual rigorous exercise, it dampens the real-world physiological gains of the training.
That is just wild.
If you are a dedicated athlete or someone whose primary health strategy is building a high VO2 max, this drug might fundamentally clash with your biological goals.
That is a massive caveat. And before we move on to how the listener can navigate all of this practically, we have to impartially review the actual physical side effects of the drug. We know the 60-year safety profile is generally excellent, but it's still a pharmaceutical. What are the day-to-day risks outlined in the sources?
The most immediate and common barrier to entry is gastrointestinal distress. The clinical data shows that roughly 20% to 30% of new metformin users experience significant GI issues, ranging from nausea and stomach cramps to severe diarrhea.
That's a massive percentage. 1 in 3 people might feel absolutely miserable for the first few weeks of taking this.
They might. The medical literature does note that these symptoms typically resolve with time as the gut microbiome adapts to the drug. It can also be largely mitigated by doctors prescribing an extended-release formulation and titrating the dose up very slowly over several weeks. But it is a very real, very uncomfortable initial hurdle.
And what about the more severe risks? I've seen older articles online bringing up a condition called lactic acidosis.
Lactic acidosis is a highly dangerous condition where lactic acid builds up in the bloodstream faster than it can be removed, leading to a dangerous drop in blood pH.
Sounds terrifying.
It is serious, but it has historical roots with a much older chemical cousin to metformin called fenformin, which was pulled from the market in the 1970s because it caused lactic acidosis quite frequently.
But with modern metformin.
The risk is incredibly rare. The sources cite the incidence rate as less than 1 in 100,000 patient years.
Oh, that's tiny.
Very tiny. As long as the patient has normal kidney function. Because the kidneys are responsible for clearing the drug from the bloodstream, lactic acidosis is generally not a significant clinical concern.
However, there is a third side effect mentioned in the sources that seems much more common and, frankly, deeply contradictory to the drug's supposed purpose.
You are referring to the vitamin B12 deficiency?
Yes. The sources state that 5% to 10% of long-term metformin users develop a significant vitamin B12 deficiency because the drug interferes with calcium-dependent absorption in the gut.
It does.
But B12 is critical for the myelin sheath around our nerves and overall brain health, right? So if this drug cleans up the cells and prevents cognitive decline via the TAME trial mechanisms, how does it also cause a B12 deficiency that actively leads to neuropathy and cognitive decline? That seems like a massive contradiction.
It is a physiological trade-off. Metformin protects the brain from inflammaging and amyloid plaque buildup by activating AMPK systemically. However, its localized effect in the gut physically blocks the receptor that absorbs B12 from your food.
The double-edged sword.
Exactly. So you are protecting the brain from one mechanism of aging while potentially starving it of a vital nutrient it needs for daily function. It highlights why this is not a magic pill that can be taken without oversight.
Okay, let's zoom out and look at the whole board here. We've got FDA regulatory roadblocks preventing official prescriptions for aging. We've got a complete lack of long-term data for anyone under the age of 65. We've got this wild exercise paradox that might sabotage your cardio gains, a 30% chance of gastrointestinal distress, and the potential to tank your vitamin B12 levels.
It's quite a list.
Hearing all of that, a listener might be feeling a severe case of whiplash right now. We started this deep dive talking about a miracle anti-aging breakthrough, and we've just spent the last 20 minutes stacking up reasons why you might not be able to, or even want to, take the pill itself.
Which brings us to the most vital realization of this entire analysis. The ultimate value of the TAME trial in this research isn't about acquiring the pharmaceutical drug.
It's not about the pull.
No, the value lies in mapping the biological pathways. Now that we know AMPK and mTOR are the master control switches for human aging, we can look for other non-pharmaceutical ways to manipulate them.
Exactly. We don't need the exact key if we know how the lock works. This brings us to the learner's toolkit. How can you, the listener, take these profound cellular discoveries and apply them to your daily routine without fighting the FDA, without risking a prescription, and without experiencing the GI side effects of a synthetic drug? How do we hack our cells naturally?
The primary target, as we established, is AMPK. We need to trick the cell into that low-power mode to trigger the autophagy Roomba, and the sources point to a highly effective, completely free, and evolutionarily proven method to do exactly that.
Fasting. Specifically, intermittent fasting.
Yes. The sources heavily reference a comprehensive 2023 biochemical review published in the journal Cell Metabolism. It outlines how specific periods of caloric restriction naturally activate the exact same AMPK energy sensor that metformin triggers artificially.
This makes perfect logical sense. When you take metformin, you are creating a chemical illusion of starvation. But when you fast, you aren't tricking the body at all. You are genuinely starving it of external energy.
Precisely. When you stop consuming calories, your circulating glucose levels drop. Your insulin levels plummet. As the cells burn through their stored ATP and convert it to AMP, the ratio shifts naturally.
The free gauge drops.
And when the body goes without food for a sustained period, the research consistently points to a 16-hour fasting window as the critical threshold for these specific benefits. The AMPK sensor accurately detects the deficit. It sounds the alarm, powers down the mTOR growth pathway, and initiates autophagy.
So by practicing a 16-hour fast, for example, finishing your dinner at 8 p.m. and simply not eating again until noon the next day, you are manually flipping the exact same biological switches that the $4 pill flips.
Exactly the same switches.
You get the cellular cleaning, you get the recycling of misfolded proteins, and you get the suppression of inflammaging. And you get it completely free, utilizing a mechanism your body was literally designed for.
It is the rhythm we evolved to operate within. Our ancestors did not have access to hyperpalatable calories every 3 hours. Their bodies relied on enforced periods of scarcity to trigger these vital cellular repair protocols.
Right, they had to hunt and forage.
In our modern environment of constant food availability, mTOR is always active. We are always growing, never repairing. Fasting intentionally restores that ancient biological rhythm.
But I want to push back on this slightly because fasting isn't a silver bullet for everyone. What if a 16-hour fast isn't viable for a listener? Maybe they have blood sugar dysregulation or a history of eating disorders, or they simply hate skipping breakfast. Do the sources provide an alternative natural tool?
They do. The sources highlight a fascinating botanical alternative, berberine.
Berberine. I've seen this popping up on supplement shelves recently. What exactly is it?
It's a bioactive, bitter-tasting yellow compound extracted from the roots and stems of several different plants, most notably a group of shrubs called Berberis. It has a profound history. It has been used in traditional Ayurvedic and Chinese medicine for thousands of years, primarily prescribed to treat severe digestive infections and diarrhea.
But wait, how does an ancient herbal remedy for stomach bugs end up flipping the same cellular energy switches as a lab-synthesized diabetes drug?
That is the beauty of modern molecular analysis. When scientists recently began isolating berberine and studying its mechanism of action in human cells, they found that it works on remarkably similar enzymatic pathways to metformin.
Oh, interesting.
Yeah, it is a potent, naturally occurring AMPK activator. It improves insulin sensitivity by increasing glucose uptake in the muscles, it lowers circulating blood sugar, and crucially, it pushes the cell into that same conservation and repair mode we've been discussing.
So it's essentially nature's metformin.
In many functional cellular ways, yes. And it is widely available as an over-the-counter dietary supplement. However, the standard medical caveat applies forcefully here. Just because a compound comes from a plant does not mean it is benign.
Very true.
Berberine is a highly powerful bioactive compound. It alters liver enzymes and can interact heavily with other medications. Anyone considering berberine as a natural longevity support must do so under the guidance of a healthcare professional.
So let's look at how the listener actually builds a routine out of this. We have metformin, for those who can get it. We have fasting, and we have berberine. They all lead to the exact same cellular destination, AMPK activation. But we have to address the elephant in the room, the exercise paradox.
Right.
So what does this all mean? If I'm using a 16-hour fast or taking berberine to activate AMPK, do I run the exact same risk of blunting my workout gains that I would with metformin?
Yes. The underlying physiology does not care whether the AMPK activation came from a plant, a fast, or a pill. If you are massively activating AMPK and suppressing the inflammatory stress response while simultaneously trying to force mitochondrial adaptation through intense aerobic exercise, you are sending deeply conflicting signals to your cells.
So it all comes down to the timing rule. This is the most actionable takeaway for the listener. If you are going to use any AMPK activator, whether it's a prescription, a plant supplement, or a fast, you absolutely must manage your timing. You cannot stack these longevity interventions directly on top of your heavy training session.
Exactly. Think about the physiological goal of the workout. If you are executing an intense VO2 max building cardio session or a heavy resistance training workout, that is a time when you want the stress signal to be loud and clear.
Let the muscles scream.
Yes, you want acute inflammation. You need mTOR to eventually kick in and build the muscle tissue. Flooding the system with an AMPK activator right before or right after that workout will blunt the hormetic stress you just suffered through.
So practically speaking, you separate them. You might take your berberine or metformin on your rest days, or on days when your exercise is limited to light zone 2 walking or yoga, where the stress adaptation isn't the primary goal.
That's a perfect strategy.
Or if you are utilizing intermittent fasting, you time your most intense, heavy workouts for your feeding window, when your body is fueled, mTOR is active, and your cells are primed for performance and adaptation, rather than deep in a fasted, low-power repair state.
You have to periodize your longevity protocols the exact same way a professional athlete periodizes their training. You purposefully cycle between periods of growth and periods of repair. You don't try to force the biological machinery to do both simultaneously because they are fundamentally opposing states.
And there is one final, crucial item for the toolkit, addressing the B12 issue we discussed earlier.
The B12 check. Whether you secure a prescription for metformin or you opt for a potent natural alternative like berberine that alters similar gut pathways, you are fundamentally shifting your metabolic absorption. Because we know that long-term systemic AMPK activation via these compounds can interfere with vitamin B12 uptake, routine monitoring is non-negotiable.
You can't just swallow a pill or a supplement every morning and forget about it for a decade.
Absolutely not. You need to actively ask your primary care physician to add a vitamin B12 serum test to your annual blood work. It is a basic, inexpensive test. If your levels start to dip toward the lower end of the range, you can easily correct it with a high-quality B12 supplement or sublingual drops.
But you have to know it's dropping first.
But you must monitor it. Protecting your neurological health is the ultimate goal of longevity science, so you must ensure you aren't inadvertently causing a degenerative deficiency while trying to optimize your cellular age.
This has been an absolutely profound journey. We started by looking at the staggering, paradigm-shifting results of the March 2026 TAME trial. We saw how a cheap, generic diabetes medication derived from the French lilac reduced the combined risk of heart disease, cancer, dementia, and death by 18% in healthy older adults.
An incredible milestone.
It really is. We zoomed microscopically into the cell to understand the ratio of ATP to AMP and how flipping the AMPK low power mode triggers the cellular Roomba of autophagy. We saw how cleaning up that debris extinguishes the smoldering systemic fire of inflammaging, and how all of this pumps the brakes on the overactive mTOR growth pathways that turn our cells into zombies.
The zombie cells are officially on notice.
They are. We navigated the murky bureaucratic waters of FDA regulations and the lack of clinical data for younger demographics. We uncovered the incredibly counterintuitive exercise paradox, realizing that sometimes blocking the stress of a workout actually destroys the benefit.
A very hard lesson for some.
Yeah, and finally, we built a practical, everyday toolkit, leveraging the evolutionary power of a 16-hour fast, exploring the ancient natural alternative of berberine, mastering the timing rule to protect our workouts, and staying vigilant with our B12 monitoring.
The overarching narrative of aging has fundamentally changed. The science we've explored today proves that aging is not an untouchable destiny or a biological inevitability. It is a measurable, distinct biological process.
It's pliable.
And because it is a process, it can be decelerated, managed, and perhaps one day reversed. But as the axiom goes, the goal of this science isn't simply to add chronological years to your life, it's to add biological life to those years. It is about extending the health span, not just the lifespan.
We want to be highly functional, vital, and cognitively sharp for as long as humanly possible. Now, as a general reminder to everyone listening, we are here to analyze the scientific data and decode the research, but we are not your personal doctors. Always consult a medical professional before starting any new pharmaceutical drug, introducing powerful bioactive supplements like berberine, or beginning a radical fasting regimen, especially if you have underlying metabolic conditions.
The biology of longevity is incredibly powerful, but it is also highly individualized. What works flawlessly for a 75-year-old in a heavily monitored clinical trial may not be the optimal intervention for a 35-year-old listening today.
So, as we wrap up this deep dive, we always like to leave you with a final thought, something that builds on everything we've unpacked today, but pushes the boundary of your perspective just a little bit further.
If you pause and think about the true magnitude of what we've discussed, the fact that a simple $4 generic molecule synthesized over half a century ago can fundamentally alter the timeline of human decay, it forces a massive shift in how we view modern medicine.
Oh, absolutely.
It makes you wonder, what else is hiding in plain sight? If metformin was ignored by longevity researchers for decades simply because it was branded as a boring diabetes drug, what other cheap, forgotten, off-patent compounds are sitting quietly in our medicine cabinets right now? What other miracles are just waiting for us to ask the right questions about them?
It really makes you look at that dusty shelf in the local pharmacy a whole lot differently. Maybe the immortality pill isn't waiting to be invented in a Silicon Valley bunker. Maybe it's already been invented, and we just haven't realized what it does yet. Keep questioning, keep exploring, and we'll catch you on the next deep dive.